Hello friends, in this video I will discuss about operon. What is the role of the operon? Where does it found in the operon? This is Mohd Asif. Let's get started. Why do we study about operon? Why necessary gene switch on and off? Each cell in the human contain all genetic material for growth and development of the humans. You know, every species has a different types of genes. It is hated or not, and when we use and when we not use the as a feed like glucose, galactose, and lactose. Okay, first we understand what is operon. Operon is the group of the gene that transcribe at the same time. They usually control an important biochemical process. They are only found in the prokaryotic species. Operon is found in the prokaryotic species. Who discovered the operon? Operon discovered the Jacob, Mohd, and Leov. Okay. Operon, they basically three genes found in the operon's elements, sugar, lactose. One of them is the gene for enzyme beta galactosidase. This enzyme hydrolyzes lactose into glucose and galactose, which is when bacteria and species use lactose, which is help to hydrolyze into glucose and galactose. Okay, adapting the environment. E. coli can use either glucose, which is monosaccharide, or lactose, which is lactose is a disaccharide molecules. However, lactose need to be hydrolyzed and digested first, because lactose has a disaccharide. It is not possible to utilize the directly lactose for E. coli. However, so the bacterium prefer the use glucose when it is needed. Okay, four situations are possible for E. coli. When glucose is present and lactose is absent, E. coli does not produce beta galactosidase. Okay, when glucose is present and lactose is present, E. coli does not produce beta galactosidase. When glucose is absent and lactose is absent, E. coli does not produce beta galactosidase. When glucose is absent and lactose is present, okay, E. coli does produce beta galactosidase. Okay, and control of the operon. When the lactose is absent, what will happen? The repressor protein is the continuously synthesized. It sits on the sequence of DNA just in front of operon, the leg operator side. You can see this gene sequence. The repressor protein block the promoter side, where the RNA polymerase settle before it start transcribing. Okay, there are three structural genes Z, Y, A. Okay, Z is it is used for Y permeates A. Z especially transcribe into mRNA directly translated protein. Z encoding the beta galactosidase enzyme, and Y is encoding the permease, which is necessary for transmembrane symporters on the cell membrane on E. coli. And A gene, what is the function of A gene? A gene is encoding the transstabilises, okay, which is basically used for lactose to allolactose. Okay, and when the lactose is present, the small amount of sugar allolactose is formed within the bacterial cell, and the fit on the repressor proteins and another active allosteric side when the lactose is present. Okay, and this causes the repressor protein to changes the SAP's conformational changes. It can no longer sit on the operator. RNA can now reach the promoter side. Okay, when the lactose is present, okay, and then what will happen? It will happen it is the change of the conformational changes in the proteins, activator proteins. when the lactose is present, and what will happen, and small proteins of the sugar allolactose is formed within the bacterial cell. This fit on the repressor proteins another active side. Okay, and this causes the repressor protein to changes its shapes and conformational changes. It can no longer sit on the operator side. RNA polymerase can now reach in the promoter side. Okay, and when both glucose and lactose are present, and what will happen? This explains how operon is the transcribed only when the lactose is present. But this does not explain why operon is not transcribed when the both glucose and lactose are present. When glucose or lactose are present, RNA polymerase can be sit on the promoter, but it is unstable and keeps falling off when glucose is present and lactose is present. Obviously, the three gene element will transcribe mRNA and restricted protein form. Another protein is needed activator protein. This stabilizes RNA polymerase. The activator protein only work when the glucose is absent. In this way, E. coli only makes enzyme to metabolize other sugar in the absence of glucose. This is the brief summary. You can see the carbohydrate, activator protein, repressor, RNA polymerase, like operon. When the present glucose and lactose not bound to DNA, activator protein, repressor left its operator side, keeps falling off promoter side, no transcription will occur. When the glucose is absent, lactose is present. Lactose is absent. Second case. Not bound to DNA, bound to operator side, block the repressor protein. No transcription occur. When glucose is absent and lactose is also absent, bound to DNA, bound to operator, block the repressor protein. No transcription will occur. When glucose is absent and lactose is present and bound to DNA, left it off operator side, site on the promoter side, a transcription will occur. Thanks for watching this video. If you like this video, please subscribe my channel and hit the bell icon. If you have any question, please ask.